and so many more. So now, without taking much time, I would like to request our resource person, Dr. Rokia Sultana, Professor and HOD, Department of Pharmacognosy and Phytochemistry, Yenepoya Pharmacy College and Research Center, to kindly start today's session with sharing her experiences on the topic. Over to you, ma'am.
Very good morning. Thank you so much, Adrija, for nice introduction. I just want to confirm whether I'm audible.
Yes, ma'am, you're audible.
Please confirm whether my slide is visible to all of you.
Yes,
Yes,
ma'am, it's visible.
no, ma'am, still yet it is not visible. Yes, ma'am,
Now
it
it's
is now
visible.
visible. Yes, ma'am, it is absolutely visible. Yes, ma'am, it is fine, ma'am.
Good morning, everyone. Greetings from Enapaya Pharmacy College and Research Center. Today I'm here to deliver a talk on recent trends in research on discovery of natural products. It's a very vast uh, topics actually, which is like a ocean. So I'll be happy if I can deliver a drop from this ocean through my talk. So I'll be dealing with this topic under this table of content, that is what are natural products and what is the drug discovery, what are the different facts about the natural products, why we need natural products for the discovery of the drugs, for natural drug discoveries, what are the advantages and what are the challenges has to be faced, how the recent advancement can help us to overcome the challenges, what are the opportunities, and finally I will be summarizing with giving few references. Now let us know what are natural products. So if you are Googling to find out the natural products, we'll get like this. So natural products, which are the compounds or the products which we get from the natural sources, which includes plants, microorganisms, higher animals, lower animals, as well as minerals. The natural products can be a product, isolated product from the from this mentioned sources, or can be also a mixture of products from these sources. Coming to the drug discovery, all of us know drug discovery has to undergo certain process, and finally we'll get a new drug to the market. So any drug we want to discover, whether it is a new chemical entity or a biological therapeutics, we need to discover through screening and so many other procedures. Once we get the drug discovered with a potent pharmacological activity, then we can go for lead optimization. Then we can study the absorption, distribution, metabolism, excretion, and toxicity studies. Finally, we can go for clinical trials. After the NDA approvals, then only the drug can come to the market, which is a known fact. And being into the pharmacy field, we all of us know it. Now I want to bring into highlight some facts about the natural products. Most of us might be knowing or some might not be knowing also. Just to revise, natural products plays a very important role in drug discovery. And if we are seeing the list of the FDA approved drugs, we can see 50% of the drugs which are either the natural products or from the natural product derivatives. Why is it so? Because this natural products has a special selectivity to the cellular targets, and these natural products also have a ligands for disease related targets, which will influence the disease related pathways, and eventually it will be able to shift the biological network from disease status to the healthy status. So this is what some more facts are. If we are comparing the conventional synthetic molecules with the natural products, we can see both advantages and challenges in the discovery process. The advantages of natural products is because of complexity of its structure. All of us know the compound what we get from the natural products are a huge molecule sometimes, which are a bulky, which is very difficult to study. Why? Because the structural analogs, structural activity relationship, which is very difficult to optimize the natural product leads, to study the natural product leads, and that can be a challenging if we are not knowing how the synthetic routes are available. Natural products are widely available in the market. If we talk about Ayurveda, we talk about Unani, they are using natural products which are available widely in the market, but the usefulness, the safety related to this, very less strong evidence is available. This is what the some facts I want to bring uh, in your knowledge. So now I want to tell why we need to use these natural products. We all know from the ancient times when the first human being has come into this world, there was no other treatment. They have taken the medicinal plants. They have taken the plants only for the treatment of diseases. And from that time, even today, the folklore medicines are the natural products. And they are used for treatment of many diseases and illness. And this discovery from the natural sources is going through a renaissance because we are so comfortable with the synthetic molecule drug discovery, synthetic molecule drug discovery is much easier. But despite of this fact, natural product derived compounds has got a much greater chemical space than those of the synthetic chemistry methods. So now, in the year of 2022, we can think of new era has come, new possibilities has come, and new disease has come. So to solve all the problems, we have to see for the novel targets, and we have to take help of the state of the art technologies. By application of this, we'll be able to do the discovery of drugs from the natural resources. As I said before also, if we are comparing the conventional synthetic methods, we can get both advantages and challenges. Let us see what are the advantages are there. The scaffold diversity and the structural complexity is one of the important advantages for the natural products. So what is the complexity is there? Obviously, the natural products atoms, very few nitrogen atom and halogen atoms are there. The hydrogen bond acceptor donors are high in number, and the partition coefficients are calculated in octanol water system, and they are also having greater molecular rigidity compared to the synthetic compound libraries. These are the parameters which helps the natural product to become to gain the importance for the discovery process. So because of this, protein-protein interactions and other advanced technologies are useful for this. I know all of you might be knowing that when we are taking a oral drug, that time the bioavailability is the most important things. If the bioavailability is not good, then the absorption will be affected, followed by other process also will be affected. But the natural products is beyond the rules of this bioavailability. So this rules of bioavailability is explained by rules which is called as Lipinski's rule, which is denoted by number 5. So this rule says the drug-like compounds which has a good bioavailability should have log P less than equal greater than equal to 5. Molecular weight should be more than equal to 500. Number of hydrogen bond acceptors should be more than equal to 10. Number of hydrogen bond atoms should be more than equal to 5. So any molecules which will violate more than one of these principles will have problems with the bioavailability. And that's why we found most of the herbal drugs, most of the drugs from the natural source, what we isolate, will be facing challenges with the bioavailability problem. Coming to the other advantages, this is optimized by evolution to serve the particular biological functions like regulation of defense mechanism, interactions with organisms, and high relevance of infectious disease. So because of these properties, they are able to uh, provide efficacy and safety. So these natural products can be a tool for bioactive compounds, which is having a wider area of chemical space, which is compared to the synthetic small molecules. I've already mentioned one challenges. Other than that, which is very common challenges we face with the natural products is availability of the source. We don't get the genuine drugs, original drugs. As a result, we have to face the adulteration. We are not knowing exact species. We are not knowing exact source. So whenever we get the alternative things, it might be the adulterated one, which is having low quality or inferior quality. Lack of standardization, the parameters of the standardization of the products, since it is a huge uh, compounds, huge molecules are present in the natural products, it is difficult to standardize. That's why most of the product which is available in the market which lacks standardization. And of course, there are technical barriers for screening of the natural products, isolation, characterization, and optimization. So as a result, the herbal drugs or the products from the natural has got a very low importance in the pharmaceutical industry, but it is gaining importance now. I want to add some more challenges which are in the beginning, which is related to mainly the herbs, that when we take
Ma'am, your voice is not audible, ma'am.
So you are saying something?
is
Yes,
it
ma'am, your voice is actually not audible.
now it is now?
Now it's fine, ma'am. Now it's fine.
Okay, fine, fine. Okay, so now I'm coming to the other challenges. When we are processing the herbs, when we are doing the extraction, we mostly heat or boil for getting more penetration in the so this may alter the dissolution rate, this may alter the environmental factors also have a great importance for maintaining the quality of the phytoconstituents. Environmental factors include soil, altitude, seasonal variation in temperature, atmospheric humidity, length of the daylight, rainfall, pattern, shade, dew, frost conditions, everything which can affect the components. So based on the different constituents, what exactly constituents we want to collect, based on that we have to select what. Suppose if we want to collect the leaves of particular plants or we want to collect the rhizomes, for example, I want to say when we are collecting the rhizomes of curcuma, turmeric, so the curcumin contains is maximum. Several surveys have said the curcumin content is maximum when it is collected in dawn, like just before the sunrise. So if you are collecting the rhizomes in before sunrise, the curcumin content is maximum amount. Whether if you are collecting in other time, the curcumin content will be low. So again, we have to keep in mind that other factors like insect planting density, competition with other genetic factors also plays an important role for maintaining the quality of the phytoconstituents. The plant collection plays a very important role. So, as I said, an example, curcumin, you have to collect in before the sunrise. Similarly, some leaves you have to collect before the flower arrives. Some fruits you have to collect before the mature state or after the mature state. So that is very very important and which is challenging. And adulteration, yes, adulteration is the main challenge. What is always done because non-availability of the drugs. Sometimes the cheap plant species is mixed, sometimes the other parts is mixed, and which has become a very very challenging for the phytochemists to do the research, because if you are not getting the proper source or proper raw material, it will be very difficult to do the research. So that is a very big point of. Next, what is the position of herbal drugs? So WHO has come with health for all, and they wanted to implement this within 2020. Based on that, prevention is better than cure. And because of health for all, the drugs from the natural source are seems to be the safest medicines, seems to be the accommodating with the human genetics, so it can be used for research for invention of lead molecules for any diseases. And multidisciplinary research on plants has been already started. Invention of the new drugs and prototype active molecules and biological tools has been done. So the position of herbal drugs has been coming up. Some examples of herbal leads which are already available in the market, for example, Google, Gymnema sylvestre, your Tucum species, papaya, ashwagandha, artemisia, satavari, mukuna, isabgol, senna, which are proved and which is
Sorry, ma'am.
available in the market. And
Madam, can you please enlarge your screen? Actually, that is not visible. Or uh, that fourth option below, ma'am.
now,
Yeah. Now it's fine, ma'am. Fine.
okay,
Thank you,
okay.
ma'am.
So these are the examples which are already using as a lead. So now I want to give highlight some on the practical outlook of herbal drug discovery, which is very known to us. What we do for this, as I said in the beginning only, when we are taking the herbal drugs for the research, the very important thing is collection, collection of the medicinal plants, because we have to select the medicinal plants based on the constituents what we are going to select for the pharmacological activity. So collection is very very important role, and while collecting the plants we have to follow, or the basically what is followed is five principles or five approaches. They are random approach. In random approach, we just go to the area and we see it. We try to observe, try to identify without having any knowledge on the plant, without having any knowledge on the taxonomy. We just pick. That is what random approach. So if we are doing this random approach, there is a higher chances that we need to take a wrong plant. Next is the taxonomic approach. Taxonomic approach is we should have a prior knowledge about the plant of interest, and we should know what is the genus, what is the family, and how we can differentiate between other species and which location it is available. So this is possible only the knowledge of taxonomy, only the knowledge about the plants. Only will be able to use this taxonomic approach for collection of plant materials. Next comes the phytochemical approach. So in phytochemical approach is also called as chemotaxonomic approach, because this is a combination of chemical as well as taxonomical knowledge. We'll be knowing about the plant all research. So in this case, we'll be able to do the proper justification with the collection. The next comes your ethnomedical approach. In ethnomedical approach, it is totally based on the information of the medicinal uses of this plant. So we'll be collecting the information. Based on that, we'll be select the last one is population managed approach. In this case, what we do is we do the survey about the plants from the local, those who are using that plants as a treatment or folk medicines. So we'll just make a survey, get the knowledge, and then we take the plants. So these are the 5 approaches are used for the collection of the plant material, and which is very, very important. And if we are failing to identify proper plant material, then definitely when we are getting the isolated compound may not be the correct one. The next step comes extraction. So extraction is nothing but what we are doing is we are extracting out the phytoconstituents which is responsible for pharmacological activity. Based on the phytoconstituents, we select different solvents and we extract the drugs. So that may be your successive solvent extraction, that may be your what you call refluxation, distillation. Based on the constituents, we'll select that particular type of extraction. Once we get the plant extract, what we do is, it is very, very important that we have to find the activity. Before finding the activity, important to find out the toxicity level. Till what level the drug is safe to use. After that, we can go for bioactivity guided isolation. We can find out the constituents, how much it is present through different chromatographic techniques. Then we can do separation and we can get the pure compound. And once we get the pure compound, again we need to check the toxicity, find out the phytochemistry by elucidating the structure, and then we can go for bioassays. So this is what in the drug discovery, what practically routinely we are following for discovery of any drugs from the natural products. Just to summarize it, we have used extraction, identification, isolation, and chromatographic method for purification and characterization. This is the must step we need to follow for finding out any new phytoconstituents which is having good bioactivity. Once we get this isolated compound, which will be leading for designing new analogs to improve the therapeutic activity or to reduce the toxicity. Finally, we can also find, check this with the economic benefit, and we can improve the quality of the product. So this process, what I told, for investigation of drugs which are very, very challenging and time consuming when we are doing in the laboratory level, traditional methods are timing. If I talk about reflux method, what happened in reflux method? Reflux methods, when we use, we don't know when the extraction is getting over because there is no limit. So we do for 15 hours, we do for 16 hours, we do for 17 hours. There is a method which is called as Soxhlate extraction. Yes, we can find out the extraction is got complete or not. But there is a limitation. We cannot use water as a solvent because this is only used for the thermolabile substance. So that is a problem when we go for the extraction method. So it is very, very important what kind of solvent we use, what methods we are used, and when we are doing the extraction, what happen is we are using the solvent which might result formation of artifacts, which might result the changing of the molecules, which can also change the nature and relative properties of the secondary metabolites. So this methods is having lot of challenges. The next step, to identify the compounds. In lab, what we use? We use phytochemical screening methods. Then we have chromatographic methods. Phytochemical screening, we all know, it is very simple method, inexpensive method, because we just use the chemical test. What we do? We just take some chemical reagent, we add it, and we see the changes, right? But this is time consuming and less reliable. Why? Because some literature says that the color will be red. So when we match the red, there will be thousands of red color. Which red it is? So and the based on the perception, colors will be changing. So it is not reliable. We cannot 100% rely on this. Yes, this is the compound. When it comes to the thin layer chromatographic technique, this is a very good technique to identify the product. We have to select the mobile phase, and the people who are working in the phytochemistry lab knowing very well how difficult it is to validate a mobile phase, because when you go to see the literature you will find, so I'm telling for carbohydrate ethyl acetate toluene 1:1 ratio will get, but I think 99% will agree with me, when you are doing we'll never get the exact result. It will be changing, ratios will be changing, will not get the proper spots, either there will be tailing, so many problems will happen. Why this is happen? Because it's a planar chromatographic method. So we'll be facing this problem when we are developing the TLC mobile phase. So advancement is required. When you go for estimation or quantification, the easily available methods are
Madam, your voice is cracking, ma'am. Uh, am I audible, ma'am? Your voice is not audible. Uh, Hello, ma'am. Am I audible?
Saura, you're audible. Ma'am is trying to clarify it.
Okay, fine, ma'am.
yeah.
Dear participants, kindly wait. Our research person had some issues related to the network. Dear participants, kindly wait. Madam is trying to fix the network. Kindly wait for 2 minutes. Sandhya ma'am, am I audible?
Yes, sir, you're audible.
Sandhya ma'am, can you please check whether madam has fixed the network or not?
Sir, no, his screen is not visible. Her screen is not visible right now.
Yes, yes, madam. Kindly wait. Actually, she is facing some issues related to the network.
Yes, sir.
Sorry, I got disconnected.
Yes, ma'am.
No
Now you
issues,
are
ma'am.
audible.
No issues, ma'am.
Yeah.
Ma'am, kindly share your screen, ma'am.
Is it
Yes, ma'am.
visible
Yes,
now?
ma'am. Now it is visible.
Okay, okay. Can you tell me till when you could hear me? PLC?
Yes, ma'am. From TLC only. The last slide was TLC. Yeah, you can start
Okay,
from
okay,
TLC.
okay, okay.
Thank you, ma'am.
Okay. So TLC it's a chromatographic technique, and which is very nice for the qualitative information. We can identify the data properly, but the technically what problem we face is 90% of the phytochemists who are doing in the lab, it's very difficult to develop the mobile phase, and the data what we get from the research articles will never be matching. So we might be knowing this will be the mobile phase, but we have to do trial and error, and then we have to develop. Because this happens because of the this is a type of planar chromatography. So that's why we'll not be getting the exact mobile phase. We have to do trial and error method for developing the mobile phase as well as for detection of the plates. Next, the estimation or quantification. To know how much amount of phytoconstituents is present, we use HPLC and HPTLC. These are the two very potential method for identification of any drugs when we compare with the bioactive marker. Using this, we can maintain the quality and the consistency of the isolate for the natural drug discovery. But the challenge is we cannot compare this to the result getting from these two methods. They are contradicting. We'll never be able to match the HPLC data with the HPDLC data. For separation of the compound, we generally what we use is column chromatographic techniques. Column chromatographic techniques, we all know it's based on different principles that may be ion exchange, that may be molecular sieving, or maybe adsorption. So it is a very useful technique for separation of any active constituents, but the problem is it is a very time consuming method, and the yield what we get is very less. Other than when we are doing in the industry level, what we do in the lab, the yield is very less. So once we do isolation, spending so much of time, the amount of product what we get will not be sufficient to do all the analysis as well as animal studies. So the isolation, again, isolation is a very challenging drug, because isolation will be able to do when we will be coming to know about all the aspects of about the bioactive compounds, whether it is a separation, identification, or characterization, and it's a very challenging job. So all the things what I have told till now, it is possible to do the natural products and what we have been doing from times, but there are so many problems. So in this the flow chart, I'm going to tell some solutions when we are doing traditionally bioactivity guided isolation for the discovery of the natural products. So here you can see I have highlighted with three different colors. One is purple color, one is orange color, one is green color. So the purple color represents the process what we are going to do for the discovery of natural products. And the problems I have highlighted with orange color, whereas the solution I have highlighted with green color. So I am starting with the natural products. That may be organism, that may be plant product, that may be animal product, that may be other natural sources. So first thing is what we are going to do is getting the crude extract, right? So crude extract, when you're using, mostly we are using different solvents. So again, based on the polarity, we are using the solvents. If you want polar compound as a constituent, we go for polar solvents. If we want non-polar compound as a constituents, we go for non-polar. Otherwise, we'll go for in between. Then we'll be fractionating the extracts for getting phytoconstituents rich extracts. Multiple round of fractionation will be able to lead the constituents and will sorry and will be able to get the single compound. And in all the steps, we are going to find out what is the bioactivity for that particular product, either in the extract stage or in the fraction stage or in the pure forms. So what problem we are facing is number one, as I tell, the availability. Availability is the first problem for either it is for the plants or for the organisms, because they are culturing into the natural habitat plants. But for the organisms, we cannot go. So what we can do is the relevant natural products are not produced when it is taken out from the natural habitat. That is the problem. So what is the solution for this? The first solution is we have to adapt new methods for culturing, and we have to adapt in situ analysis. Solution for the problem 2 is when we are doing for the new methods, we have to take other level, that is that molecular level. We have to induce some expression of genes, and we have to study. So then it is possible to find out at what condition, at what environment we'll be able to culture. So that will be very helpful, and we'll be able to get the natural products how we are getting from the natural habitat. The problem number 3, this we are getting when we are taking the crude extracts. We don't know whether the natural products, whether the phytoconstituents, what we want to have, whether it is present or not. So it is not known to us, because the crude extract we have taken, if we are the lead compound, what we want for the therapeutic activity, if it is not present, it is of no use for the further process. So the problem number 4 is the natural products, whether it is having drug-like properties or not. That is what I told you, the rules where the bioavailability concerns. The problem number 5 is whether the natural phytoconstituents is present in sufficient amount or not. So these are the 3 problems which is a very difficult to find out whether it is there or not. So what is the solution for this? We have to adapt new method for dereplication. We have to adapt new method for extraction, pre-fractionation of the extract using the newer technology. Problem number 6, when we are going to the single compounds, it is very difficult to find out what is the targets for the natural product using the genetic level. So for that also, we have the solution. We can find out the compound based on the mode of action, elucidation. We can take help of artificial intelligence, and we can solve this problem. So this is the sum of the problems. Later I'll be explaining some more challenges. So there are certain approaches has been given to improve the natural product drug discovery. There are so many. I have listed out few here. They are top-down approaches, bottom-up approaches, diverse sampling and culturing, and high-throughput screening. So what is this top-down approaches? So I'll just show you the flow diagram here. You can see in one diagram only I'm showing both top-down approaches as well as bottom-up approaches. So top-down approaches is coming from up to down, whereas bottom-up approaches is coming from down to up. In top-down approaches, we are starting from the source level, and then we are utilizing the source of the things without having prior knowledge of the molecular level or the genetic level or the enzymatic level. So we are not associated with what are the genes or what are the enzymes are responsible for biosynthesis of the product. That's
Madam, your voice is not audible.
is what
Uh, Madam, can you hear us? Your voice is not audible, ma'am.
am I audible now?
Yes, ma'am, you are audible. Kindly make the large screen, ma'am.
All right.
Uh, yes, ma'am. Now it's fine, ma'am. Thank you.
So I was talking about top-down approaches. We start with the organism, the resource from the source, and then without having scientific knowledge or enzymatic knowledge, which is responsible for the biosynthesis of the active constituents, we go for production of the natural products. Whereas in the bottom approaches, what we do first we identify what is the gene which is responsible for biosynthesis of the products. Manipulation technique, reverse transcription, translation, and then we go for synthesis of the natural product, and which is systematic approach. So here we can see this is what we do. We just take the drugs. We know this is the phytoconstituents. We take the sources and then go for other methods, and directly we go for isolation of the product. Whereas the bottom approach is where we take the host, study the genetic as well as enzymatic level, what is the mechanism for the products, how it is biosynthesized, and what is the product it is going. Then we take help of different genetic methods to improvise the products, and then finally we get the products. So this is bottom approaches is much more advantageous than the top approaches. And one of the methods which is used for the top down approaches is metabolomics. So metabolomics, we already know that the plant or the natural products are having a very complex molecules, very big molecules with a very high molecular weight. So this is not possible to study properly, and that's why the techniques which is called as metabolomics, where what we can do is we can study a large number of metabolites which are present in the biological compounds. So here what we use is high throughput analytical chemistry, multivariate data analysis we use to study the metabolic mechanisms. So what we do is we take the entire set, complete set of metabolites. Metabolites means the product which is present in the drugs and which is responsible for metabolism. So this metabolomics study, we get the complete knowledge about the biological of the end product what we are going to produce, and the complete knowledge will be given. So in fact it can be called as a snapshot of the physiology of that cells. So from the cells, we'll be able to know what type of and what is their nature in a molecular level. So that is what we are studying in metabolomics. And metabolomics is having a wide range of applications. It can be used for pharmacology, drug treatment, dose evaluation. It can be used for toxicity studies. It can be helpful for biotechnology, crop improvement, transgenic research, plant breeding, systems biology, dynamics in biological system, enzyme discovery, discovery of biochemical pathways, link between the metabolites and the activity. It also can be helpful for microbial food technology, nutrigenomics, and very importantly, it can be helpful for human disease diagnosis. It also can be used to find out the biomarkers discovery for any kind of risk determination also. And its very important role in pharmacological drug discovery and treatment. So this is what about the metabolomics. You have another methods which is called as reverse pharmacology. I hope you have heard about this. So Sir Ramnath Chopra and Ganesha, they have used these terms in the first reverse pharmacology. And using this, they have documented many Ayurvedic drugs which are clinically used. So this reverse pharmacology, it will relate. It was like before it was laboratory to clinic. So what we are doing is first we are discovering the drugs and then we are going for the treatment. So but this reverse pharmacology will be act in a opposite direction. First we'll know about the disease, then we'll be targeting, and then we'll come to the isolating of the compound. So that is what is reverse, and as a result, here in reverse pharmacology, the first importance is safety, later comes the efficacy. So I just want to compare the conventional methods of drug development with the reverse pharmacology of the drug development. So in this, the first one, it is showing the conventional method where you can see you are targeting the identified compound, then you are going for lead identification, optimization, preclinical study, clinical study, and then FDA approvals. So all of us know the duration, what it takes for the drug coming to the market. The discovery phase takes a huge time. After that, the development phase takes nearly about 10 to 15 years, and the money what is been invested is around 1 to 1.5 billion US dollars. Whereas the reverse pharmacology, we use the traditional knowledge database, which are already used for clinical use and safety. And then based on that, we go for extract and formulation. We do the fluorescence using output screening method, and then we go for preclinical studies. Then we go for clinical trial and FDA approvals. So the discovery phase has reduced. Whether since the discovery phase has reduced, the other part also is reduced. You can see discovery, the development phase has reduced to 5 to 6 years. As a result, the investment also has become down. So the reverse pharmacology for the traditional medicine is faster, economic, and safer compared to the conventional methods. And very, very important is application of artificial intelligence. Most of the challenges are being solved when we use the artificial intelligence, because we'll be able to identify the compounds and drugs either by modifying the compound or by studying the target molecules. I'm just giving one examples of this artificial intelligence, which is an in vitro modeling. It's called as human-induced pluripotent stem cells. So this is actually we I have found a very recent article where they have used this in vitro modeling for using the cells for making the cells which are having functions in the neurons, especially for the neuron disorders or the nervous system disorders. They are using, what they are doing is they are taking a different somatic cells, and that can be potentially converted into the neuronal cells. So I have given you the here. So what they are doing is high throughput screening. They are using, and they are using a chip, microfluidic device, and they are able to convert any kind of somatic cells into a neuronal cells. And as a result, it is very easy to study the drugs which are having disorders related to the brain. So what they are doing is they are searching the database, what are the compounds are present, and they are able to find out the lead molecules. Then they are undergoing the high throughput screening using the 3D cultures, and then they are able to get the drugs which are responsible for the neuronal disorders. So this is one very good the tools which is helpful for the studies of neurovascular disorders. Next comes other analytical tools, what made the natural product drug discovery process a very smooth. I'm talking about here some strains of bacterial strains from the natural sources is been taken, and then that is done the profiling, image-based phenotypic profiling. And the metabolomics is studied based on LCHRMS. Integration is done, and then this is further studied by LCMS studies. So here, the compound which is isolated is quinosinomycin, different compound A to D, and it is a very good and very advanced way method to find out. And that compound, which is fungal activated clusters, which is generated and from different species, and then it is predicted with well-known homologous, and then it is metabolomic studies are done, and it is characterized by LCMS and NMR analysis. So in this, the advanced analytical technologies has implemented, and which is given smooth running of the discovery of natural product. One of the example is fluorescent-based live cell analysis. So in this method, what we will be able to see is how the different cell patterns, cell viabilities, how it is related to the stress, how the products are responsible for the stress. So especially this kind of in vitro method is being used to study the stress related disorders. And this is what one more advanced method is column chromatography or the size exclusion chromatography. Then we get the compound, and then we send for NMR, right? So in this case, it is automatically, it is related, so it is also helpful and also able to come out from the challenges. So I have told about the problems, difficulties, and the solution. So now let us know what are the opportunities are there in the field of natural products. It is leading to the extraction methods. All doing extraction, right? So we are getting screening thousands of compounds. All of us are doing, when we want to screen certain compounds for a particular kind of activity, we need to screen thousands of plants at one go, which is not at all possible. But if there is a extract library, if all of us we are able to make the extract library, what is the method of extraction, what type of compounds we are getting, and what are the challenges and how to solve this, if you are able to build it, then I think the process will be very smooth. And I am happy to inform you that Nicholas Piramal India has built up a plant extract library which contains 6,000 extract which is prepared from 2,300 plant species from India. So every one of us, if we try to build this kind of extract library, or locally or between the colleges or between the universities, those who are doing this plant related research, we can have a better knowledge about the extraction process, and we can overcome the challenges for the extraction. And as I said, the drug discovery process is going through a renaissance. We have spent many decades in the shadows of synthetic drug discovery. Now it's a time so we can take the opportunities, the advantages of the natural products, and we can bring the natural product research forward. So WHO has given this that by 2050, all the major health problems will be solved by the traditional system of medicines in collaboration with modern technologies. So the major health problems like cancer, hypertension, diabetes, malaria, viral, dengue, chikungunya, and others. All this is possible if we all are start giving more importance to the research on natural products. To summarize my talk, uh, we are knowing that herbal drug development are growing interest, and we have to see that it should have a minimum side effects. We should be able to explore the medicinal and biological properties of the previously natural product which is already which is not done. Along with this, we also need to focus on visualization and identification of unused herbal plants over the world. What is people are not used. Also, we have to give a historical look to the previous discoveries, and we have to see how the discovery process has faced the challenges, and we have to compare with the current status. We have to give the future opportunities, and so that we'll be able to come out with the challenges, and we'll be able to discover new drugs to treat different disorders. As I said, the natural product contains the complex chemical structure, and which is a very advantageous for discovery of the drugs. So the world is gifted with nature, and the man is gifted with brains. So let us make use of it to discover the new entities which will be available to the common people in economic rate, and will be happy to lead a good life on this earth. So these are the few references I have mentioned here. Thank you very much. The session is open for the questions.
Thank you so much, ma'am. So I would like to thank you again, once again, ma'am, for giving such an elaborative and informative session. I hope all our participants have got more clear vision on the field of discovery of natural products. We have few questions based on your session, ma'am. So if there is a permission, I would like to take the questions for the session.
Yes, sir. Yes, sir.
Okay. Ma'am, from Nobarun Mukhopadhyay, we have two questions. The first question is, what are the methods that are generally followed for structure modification in natural-based drug discovery?
Yes, Navarone, it's a good, very good questions. Usually, what we use for the modification, first of all, I told the main problem for the natural products is the bioavailability. Most of the drugs are not following the Lipsinski's rule. So as a result, the bioavailability is the main problem. So what we can do, we can take help of the artificial intelligence to modify the drugs. We can also take help of novel drug discovery methods, targeted drug discovery methods, and we can do the modification. I hope I've answered your question.
Ma'am, Nobarun has one more question. Uh, the question is, can traditional literatures like Ayurveda give us idea for selection of lead molecule in natural product-based drug discovery?
Not alone Ayurveda can give you. We have to, as I said, you have to have this five approach. Out of that five approach, the first one is which is random approach. Random approach is not at all possible. So you can have the surveys in the local areas. You can also have the ethnomedicinal database. You can also have the taxonomic knowledge as well as the phytochemical knowledge. So combining this, you can have a lead molecule. Yes.
Hope so. Nobarun Mukhopadhyay sir has already got his answers. So moving to the next question, ma'am. Sindhu Priya ma'am has asked one question. Some biological activities of extracts are resulted because of synergistic activity of active components. This is the Ayurvedic approach. So is it really necessary to isolate individual components from all extracts?
It's based on your research of interest. If you want to have a pure compounds for your extract, like synergistic in the extract, you have two compounds. Sometimes it will be potentiating, sometimes it will be diminishing the activity of each other. So you have to see where how it is affecting. In case if it is synergistic, there is no need to isolate, but in case if it is diminishing the activity, yes, you have to isolate.
Okay. Hope so. Sindhu Priya ma'am has got the answer. So moving with the next question, ma'am. Sandhya Jaishwal ma'am has asked one question. Any free accessible software which we can utilize for natural products?
Free accessible software. Actually, some softwares are available which will be on what you say trial basis. So that you can use once you become comfortable with that. You can have the license for that. And there are certain institute who has the already made database, that is FRLST. They are doing a tremendous job on making the database. So you can go to their website. You will get each and every details of the plant products.
Okay. So our next question is Sudarshan sir has asked how to identify novel phytoconstituents from any herbal plants.
How to isolate? Isolate, you have to find out what kind of constituents you're looking for. You have to select the method of extraction. Then you have to go for identification by using TLC. But if you are knowing the compound, you can also target the what you say is in silico studies or molecular docking. You can target the proteins which are responsible for that particular activity, and after that you can do the GCMS or LCMS to find out what are the compounds are present, and then systematically you can go for isolation. You like this you can isolate the product.
Okay. Our next question has been asked by Neha Vijay ma'am. Ma'am is asking which is the best method of extraction in our lab.
Best method of extraction what we use is if you have thermolabile substance, we go for oxidation method, where we'll be able to know whether the extraction is, and if it is water, then we go for refluxation method. But the disadvantage is we'll not be able to know whether it is extraction is completed or not. So you just have to do for more hours to get the extraction done.
We have participants Ashwathi Hate. Ma'am, ma'am, you can unmute yourself and you can ask the question to our speaker, ma'am.
Good afternoon, ma'am. Am I audible?
Yes, ma'am.
Hi Ashwati, I'm happy to see
Hello,
my
ma'am.
student is here.
Hello, ma'am. ma'am. It's actually a pleasure to have listened to you after so
Can you
long.
hear me,
First of
Aswati?
all, I want to. Yes, ma'am, I can hear you. It's a pleasure to have listened to your talk. And
Clear?
first of all, I want to congratulate you for this wonderful presentation that you have given.
Yeah.
Okay, ma'am. I just had one question. Most of the herbal formulations are basically polyherbal formulations, if I'm not wrong. So they contain more than one
Hello?
active constituent. Can you hear me, ma'am? Are you able to hear me?
Hello?
Are you able to hear me, ma'am? Can you hear me, ma'am?
Aswati, you're not audible.
Uh, can you hear me now? Hello? Hello? Can you hear me, ma'am? Hello?
Um,
Saurabh.
Hello? Can you hear me, ma'am?
ma'am, Ashwati, ma'am,
Are you able
ma'am,
to hear
maybe
me?
due to some network issue, our speaker is not able to hear you. If you can kindly drop the question in the chat box, it will be very helpful,
Okay,
ma'am.
fine. Sure, ma'am. Sure.
Yeah. We have Saurav Mohantu sir. He also wants to ask some question. Sir, you can unmute yourself and ask the question.
Thank you so much, ma'am, for giving me this opportunity. Well, first of all, I would like to thank Rokia, ma'am, for this celebrative session. Thank you so much, ma'am. You have explained a nice way. Basically, ma'am, my question is something which is related to pharmacognosy, natural products, and as well as drug delivery system. So, ma'am, the thing is, whenever we are extracting or isolating any compounds, after that we cannot take that compound for the drug delivery process. We have seen several natural products or uh, compounds which is available. People have extracted, isolated very successfully, and they have only taken those compounds for few drug delivery systems. uh, so on that note, what is your point of view? I mean, like after
So,
doing all of this extraction, isolation, doing all this analytical procedure, even we cannot take that origin or original compound for any drug delivery process, or we cannot deliver via any drug delivery. Then what is the use of those all these processes? Am I audible, ma'am?
Saurabh, can you repeat your question? I was not able to hear. Now it is okay.
Yes, ma'am. My question is, ma'am, after isolating, we are getting so many successful compounds. People have isolated those compounds. But my question is, we cannot simply take those compounds because some of the compounds they have the issues with the hydrophilicity or lipophilicity, bioavailability, kinetic profile, etc. So we cannot directly take those isolated compounds for the drug delivery system. So, what is the use of doing all this process? Then after that, again we need to modify that compound. That means again we need to add some other substitute so that we can improvise the property, chemical property or physical property of the compound. So, what is your opinion on
Yeah,
this
yeah,
note?
yeah, Saurabh, I got your point. I got your point. Yeah, so that's what we are doing is top to down. That is what I wanted to show in that slide. We are finding out we are without knowing about the constituents, we are going for isolation. And then when we isolate the compound, we are not knowing or not able to modify it. So the recent approach, what is there is bottom to down. First, only you find out the constituents. You go for the genetic manipulation. You go for the other method of manipulation. Make the product which is readily available and which is able to treat the disorders properly. Then go for isolation. So that is what is the recent approach. We should go for bottom to up, not top to bottom.
Thank you, ma'am. And
Oh,
another doubt, not doubt actually, another query I have actually, ma'am. There are several people who are preparing
Yes.
some natural products and they are utilizing more than 50, 60 compounds in a single product, and they are claiming that it can improvise your health condition or maybe liver condition or something like that. So on that aspect, what is your note, ma'am? What is your thought? Whether those products will be really successful in the market?
Actually, Saurabh, what happened is mostly the herbal drugs will be having multi components. In that, some of the compounds will be having the synergistic effect, and they will be potentiating the activity of one compound to another compound. If that is the case, in that case, it will be helpful. But if the some compounds is having the which will be inhibiting the activity or reducing the potency of the compound, in that case it will not be fulfilling the value. Sometimes it is very very important to know whether it is having a synergistic effect or not, and then only we can give such kind of formulation. So research is very important to do in this field. What is available in the market, it is not safe what is being told as a safe. So it is always, and also there will be interactions. There will be the compound to compound interactions will be there, which can be fatal, which can be very very toxic also.
Yes, ma'am. That is a note because during our COVID time, there are so many people who have introduced some natural based products in the market, and in those products there are more than 30 compounds at a time. So my first thing is like it is really difficult to isolate those compounds again to check the individual activity, and in together if I'll mix all those compounds in a single base or single platform, then what will happen, whether it will really give synergistic effect or after administering to the body maybe it will have some other effects. So that is my actually my query.
Yeah, if it is giving synergistic effect, then it is good, but at some time it will be toxic also, because at a particular dose, most of the herbal products at a particular dose it acts as a medicine, and when it crosses its limit, it will be as a poison. So this is very concern that very difficult phase. Like it might lead to some toxic effect. It might damages some organs. That can happen. So it cannot be given like this blindly. Given is very dangerous.
I think so. Saurav sir has cleared all his doubts. Uh, let's move on with one more question, ma'am. There are
Yes,
some
thank you,
questions
ma'am.
from the as well, comment box. So we have some questions from there. Uh, the question is, in which phases of natural product discovery we can use AI?
Actually, AI we can use when we know what activity we want to do. When we are able to select the therapeutic activity, that moment only we can take help of AI, and then it will be very useful. Like we will be based on the activity, we'll be able to target the constituents. Once we know what constituents is responsible for it, and AI will give a choice how it is synergistic, or whether it is having any effect or not. So when we are able to target that molecules, and we are able to find out what is the constituents responsible for pharmacological activity, it will be helpful. So I think when you are able to select what kind of activity you are using and what is your drug, and do the extraction, from the extraction step uh, onwards you can take help of the AI.
Okay, ma'am. There is one more question. What is the logic behind using more than 50, 60 natural compounds used in one product? What is the logic behind of using more than 50, 60 natural compounds for one product?
uh, I think the same question what Saurabh asked, same question. So again, I am going to tell 50, 60 compound herbal drugs will be having. So it is not safe to use until unless you know you cannot go for 50-60 compound or 30-40 compound. You can use the phytoconstituent rich fraction, which may have some 4-5 compounds, and each compound may be having synergistic effect or no effect on the lead molecules. Then only we can use. If there is some inhibitory effect or other effect, then it is dangerous. So we have to study thoroughly the phytoconstituents, the nature of the phytoconstituents at compound level as well as molecular level. Then only we can combine. Otherwise, it is not at all advisable.
There is one question in the Meet chat box, ma'am. Is there any Indian medicinal plants database that are comparable to Chinese medicinal plant databases?
Yes, there are a lot of plant databases are available. FRLST has created, NCBI has created, Medicinal Plant Board has created. There are a lot of database. You can get lot of data from there.
Oh, okay, ma'am. One more question. Ma'am, Soxlet extraction is better technique or microwave assisted extraction for extracting phytoconstituents is a better technique?
Method wise, microwave assisted extraction is better techniques, because within a span of very short time we can get the extraction done, as well as yield will be very much higher than the Soxhlet method. But the problem is, in microwave assisted extraction, you will not be able to do more amount. You cannot take the more amount of drugs. The maximum amount of drug what you can take is 100 grams. So if you need your extract more, then you cannot utilize microwave assisted extraction. So yield wise, Soxhlet is better, whereas the time concern and the extraction method, so microwave assisted extraction techniques is better.
Is it possible to isolate the constituents in hero mineral compound?
Hero mineral compound is not clear to me.
Okay, ma'am. Ashwati ma'am is again raising her hand. So, ma'am, you can unmute yourself and ask the question.
Hello, am I
Yeah,
audible, ma'am?
you're audible.
Okay. Uh, okay, ma'am. Hello. So first of all, I want to congratulate you for the wonderful presentation that you have given, ma'am, and
Thank
hope
you,
you're
dear.
keeping well. Uh, Ma'am, I just want I have herbal formulations are basically comprising of more than one active constituents, right? So in such cases, is there any specific, say, analytical method or any method which will distinguish these constituents, or is there any foolproof method to separate the constituents?
Aswati, can you repeat? I think your voice is breaking, so I could not catch your question.
Ma'am, Ashwati ma'am is having again a network issue. The previous question that was asked, that was, ma'am, is it possible to isolate the constituents in herbo mineral compound?
Herbo mineral, you mean to say the combination of mineral and herb? It's not, still not clear what exactly the question is.
Yeah, ma'am. Mineral or herbal based with herbs combination.
You mean to say that you want to do the formulation?
Yes, we are usually using these preparations in Ayurveda, where we are using different metal and mineral based basmas with herbs.
Okay, okay, okay. Fine, fine. So you are combining. Actually, we also do some kind of formulation, which is called as nano formulation, where we can use the minerals like gold, silver. Sometimes we are using carbons. So that can be used, but we have to see that it should be compatible with your heart. There should not be any interactions, or it should be having any toxicity, because mostly the minerals will be having toxicity. So the toxicity level has to be in consideration. You can go for formulation.
Okay. One more question, madam. Will this mineral or metal change the this herbal this composition in any way? Will it
Yeah,
affect?
that's what I told, that they should be compatible. Like it should not affect the nature of the phytoconstituents. Like if it is reacting with the phytoconstituents, then you will get the derived product derivatives. You will not get the actual phytoconstituents, and that is the problem. So you have to see that the drug should not react. The metal should not react with the drug or the constituents. So if it is inert in nature, or the interaction is not there, then it is possible.
Okay.
Yeah, Ashruti.
Yes, ma'am. Ashwati ma'am has written her question. Which is the best suited analytical method to separate the constituents of a polyherbal formulation?
Ashruti, now there are many methods available, and then now you can see the combination combined methods are available. Like you can get what I have given one example, that the column chromatography is fixed with the NMR. So like that you have HPLC, which is fixed with LCMS. So what we'll do is you'll be able to separate, at the same time you'll be able to see the constituents. So this the advanced analytical methods are very helpful. What we can find out for a polyherbal formulations. But if you talk about laboratory level, so you can go for column chromatography only, and then you have to see what kind of constituents it is, and then you have to choose what is the principle of that column chromatography. Based on that principle, you can go for separation method. I hope, Ashruti, I have answered your question, and you can be in contact with me in case of any help. I'll be there to help you. Don't worry. If you are establishing any method, analytical methods for separation, I'll be there to help you.
Ma'am, we have Kanu Anna, ma'am. You can unmute yourself, ma'am, and ask the question. Okay. Hope so. So all the participants are requested, if you have any kind of more queries from our speakers, then you can kindly mail to the mail ID, and the speaker will be replying to all the questions. So once again, I would like to thank you, Dr. Rokia Sultana, Professor and HOD, Department of Pharmacognosy and Phytochemistry, for such an informative session, and hope so all the doubts and the confusions that our participants had been cleared. Thank you so much, ma'am. And it is for the kind attention for our participants that today we have ended up with this only single session. So for the afternoon session, we do not have any session for there, as already informed in the beginning of this session. So the feedback link has already been posted in the chat box and the comment box. Kindly fill up the feedback link and to get the proper attendance for the same. And thank you so much, all of you, for joining for today's session. Hope to see you all again tomorrow for the tomorrow's session. And thank you once again for this wonderful session, ma'am. Thank you so much.
Thank you, Advija.
Thank you so much, ma'am, for joining with us. Dear participants, kindly fill the feedback form, which is already posted in the chat box as well as YouTube comment box. Thank you so much for joining with us. We'll see you tomorrow. Thank you. Dear participants, you can already see ma'am has shared the email ID. Once again, I'll share with you so that if you have any further queries, you can mail her. She will be happy to help you. Here is your mail ID of Rokia ma'am. You can always communicate with her. Thank you, everyone.